Okay, so good afternoon, everyone.
Good
I hope
afternoon, sir.
I hope I am audible as well as my screen is visible to all of you.
Yes, sir.
Okay,
Yes, sir.
so we stopped here yesterday. We were discussing about that how the neurotransmission process actually happens and what are the factors which control this process. So, in short, if you look at it, we actually uh, throughout the course of our understanding of the nerve cell function, we have actually migrated from the area where the nerve cell is located from that point to the architecture of the nerve cell, and finally we have found out that how the neurotransmission process actually happens. And it essentially happens in most of the neurons by means of the chemical synapses that we have talked about. These are dependent upon the chemical signals, which are commonly referred to as the neurotransmitters. So we talked about that how the neurotransmitters are actually released in the synaptic cleft, and what is the fate of the neurotransmitter is decided based upon that what kind of receptors are present in the postsynaptic membrane, and depending upon the kind of receptors the binding of the neurotransmitter to the receptor brings about the changes in the sense that it can either open the ligand gated channel as it has been shown over here if the receptor is an ion channel itself, and how the signaling is controlled here is dependent upon what kind of the neurotransmitter you're dealing with. If it is a simple neurotransmitter, if you don't need much of an energy to synthesize that neurotransmitter, like we yesterday talked about acetylcholine, this neurotransmitter is not very difficult to synthesize. It consists of two parts. One is the acetyl group and another is the choline group. So you can very easily synthesize both of these components and you can recycle it back by means of an enzyme which is called as acetylcholine esterase that breaks down acetylcholine into acetate moiety and the choline group, which are taken up back by the presynaptic membrane by means of certain transporter proteins. And once you take them back inside the neuron, then it is recoupled in order to synthesize acetylcholine. That's simple. There was other consideration that if the neurotransmitter is difficult to synthesize, like for example, a molecule that is dopamine or serotonin, these are the neurotransmitters which are difficult to synthesize. And hence, it doesn't seem logical that you have or you deploy uh, a neurotransmitter degrading enzyme in the synaptic cleft to control the activity of neurotransmitter. Instead, you can work out a way by the way of which there are certain transporter proteins which have a great degree of what you can say affinity for these neurotransmitters, so that as soon as they are released into the synaptic cleft, there will be a competition for dopamine binding to the receptor on the postsynaptic membrane or to transport it back into the presynaptic cell. So these are the ways in which the activity of neurotransmitter is kept under control. You don't want that the activity of neurotransmitter should last for a long duration of time. So depending upon now how the neurotransmitter is or what kind of a neurotransmitter you're dealing with, the one thing is for sure that the neurotransmitter is going to decide what is going to happen with the postsynaptic cell, right? This much is fine.
Yes, sir.
However, there is an interesting aspect here. Although you have the same neurotransmitter, depending upon the type of the receptor also, the fate of the neurotransmitter, in the sense that what will be the repercussion when neurotransmitter is released in the synaptic cleft, that is decided. Meaning, like for example, we talked about that if say suppose acetylcholine binds to ion channel kind of a receptor, then binding of acetylcholine opens the ion channel and causes depolarization of plasma membrane of the postsynaptic cell, and that leads to generation of another action potential that we talked about, that there could be generation of another action potential if this is the situation. However, there are other ways of dealing with acetylcholine as well. Like one we have already discussed in the neuromuscular junction. In the neuromuscular junction, there could be a different way in which acetylcholine presence is perceived, as well as by different neurons, acetylcholine can be perceived differently. So how it can be perceived differently, it depends upon two different what you can say sensors or receptors of acetylcholine. One kind of a receptor is referred to as muscarinic acetylcholine receptor. Another kind of a receptor is referred to as nicotinic acetylcholine receptor. Now, the basic difference between the two is nicotinic acetylcholine receptor is basically a ligand gated channel. So if you want to distinguish, this is basically a ligand gated ion channel that we have discussed in the last class itself. However, muscarinic receptors are not channels. These are actual receptors, meaning something like hormone receptors. So they are coupled with some other, what you can say, these molecules which are depicted over here are commonly referred to as the second messenger molecules. So it depends that which kind of receptor acetylcholine is going to bind, whether it is muscarinic acetylcholine receptor, then the kind of signals it is going to send will be different. Now you can see the effector molecules, what are going to get affected by binding of acetylcholine here. However, in the nicotinic acetylcholine receptor, you can see that how different is the mechanism. It's just going to open the ligand gated ion channel, and you can get you can make the ion to go for an entry depending upon the presence of the ligand, which is nothing but acetylcholine here. So one basic difference is whether acetylcholine is going to bind to muscarinic receptor or whether it is going to bind to nicotinic receptor. That will decide what kind of a fate is going to happen. Is this much fine?
Yes, sir.
Okay, good. But even when it binds to the ion channels, still the fate, what it is going to lead to, might be different. Okay, now if I say this, it becomes slightly more complicated to understand, right? So we will look at it in this manner. Okay, now this is the area which is a ganglia. Ganglia are, do you remember what are the ganglions? We talked about this when we were discussing the anatomy of the nervous system at the beginning. It's actually not in your course, but it's going to be very helpful for remembering that what is going to happen with acetylcholine. So do you remember what are ganglia or a ganglion, if it is, if you want to say it in a singular fashion? Yes, do you remember?
Sir, they are collection of cell bodies.
Yes, collection of cell bodies, one important thing, but collection of cell bodies outside the central nervous system, that is more important. Okay. So when we are talking about ganglia, these are collection of cell bodies, but they are outside the central nervous system. Now just look at this. This is just a representation. Now you can see this cell body. Okay. You are not going to consider this neuron to be a part of the ganglia, right? Because its cell body is out. Its cell body is in the central nervous system. This is just a depiction. This is the brain and this is the spinal cord. Okay. I hope that you can see my cursor. This is the brain and this is the spinal cord. Okay. Now, the neurons which emerge out from the brain, they can be of two different types. One neuron could be a neuron which is going to get associated with the parasympathetic nervous system. Okay. And the parasympathetic nervous system is going to have its neuron getting into the ganglia. And what exactly happens here is that ganglia is going to connect the neuronal axons which are coming from the central nervous system to the cell bodies or the dendrites of the neurons which are going to which are going to go to the peripheral organs or the peripheral nervous system. We'll talk about how it is actually seen. So if we take an example of the parasympathetic nervous system, then what is actually going to happen is that the neuron emerges out from the brain. Its axonic terminal, the axonal terminus of this neuron is going to get uh, involved with another neuron's dendrite in the ganglia. So there will be several such neurons, right? In the ganglia, they form the synapse here. And now, if you secrete acetylcholine here, then N indicates nicotinic acetylcholine receptor. So what will happen here is that it will make another neuron fire, okay, and release another acetylcholine molecule, which will bind to muscarinic acetylcholine receptor. It's slightly complicated, right?
Yes, sir.
So what do you think? What will happen? See, what is happening here, what is interesting to note is that you are releasing acetylcholine from the first neuron, which is coming from the central nervous system, right? This acetylcholine is going to excite the next neuron to again release another acetylcholine molecule. Are you getting the point? But this acetylcholine will cause another effect because it is binding the muscarinic receptor. Okay, now what is that effect? We will talk about that slightly later on because I don't want to complicate the matters right now for you. So let's take a simple example. Okay, now in the previous slide itself, we have seen that there are nicotinic acetylcholine receptor and they are of two types. One are alpha types, which will contain only alpha subunit, and another are alpha beta type, which will contain alpha as well as beta subunits. Now, what are these subunits and what kind of mechanism is associated with it? It will be pretty clear here. Okay. Now, see, in this another example, you can see that the neuron which is coming out from the central nervous system is going to secrete acetylcholine at its synaptic cleft, which is in association with another post synaptic neuron coming in the ganglion, but it is having a nicotinic acetylcholine receptor. So this neuron will also fire, right? There will be an action potential generated, but this neuron is going to secrete a hormone which is called as norepinephrine. Is basically another alternative form of epinephrine. Epinephrine, you know what is it?
No, sir.
Adrenaline.
Yes, adrenaline. We know that in case of excitement or in case of fear, in both these situations, we get what is called as adrenaline rush. This is not adrenaline, however. This is called as noradrenaline or norepinephrine, which is another alternative form, which has a different role to play. Okay, this acts on the alpha beta kind of receptors. These are different receptors for norepinephrine. These are not for acetylcholine. Okay, and they cause an effect on heart and blood vessels. Okay, now what kind of effect is going to depend upon what kind of a signal is being sent. But essentially, this leads to what is called as vasodilation. Like say, for example, I will give you one very simple example. Now, this is associated with sympathetic nervous system. So the understanding goes like sympathetic nervous system has some sympathy for your visceral organs. Visceral organs are internal organs. So in case you have some kind of fear sensation, have you ever noticed this? In case you are having some kind of a fear sensation, then you tend to have some kind of tingling sensation in your tummy, right? Have you ever experienced this?
Yes, sir.
Yes, sir.
Okay, and that is because your sympathetic nervous system is playing a role. This is going to kind of shield you from any kind of stress or fear. Okay, so what is going to happen is that there is vasodilation, and what actually happens is the visceral organs tend to get more blood supply. This is basically to protect the visceral organ from some kind of a stress. Okay, that can be any stress. I am not going to go into the details of it. Okay, another mechanism is associated with another acetylcholine receptor, which is associated with another sympathetic nervous system. But this acetylcholine receptor is adrenergic because it is going to respond to adrenaline or noradrenaline, right? But this kind of a sympathetic receptor is going to respond to cholinergic kind of a system. Cholinergic system is basically associated with acetylcholine. So one acetylcholine molecule released by the presynaptic neuron. It gets connected with the receptors on the dendrite of neuron present in the ganglia and causes this neuron to fire. Now, when this neuron fires, then look at it, what is happening. It is actually going to make this neuron secrete another acetylcholine molecule, but now it is binding to muscarinic acetylcholine receptor. This is again associated with sweat glands and vessels. What this is going to cause is this is going to constrict the vessels. Now you don't need to actually remember what is going to be the response. Okay, you just need to remember this much, that it is same acetylcholine molecule, right? Seems to be a same kind of a neuron, but it is the receptor which is going to be the deciding factor, that what kind of a response you are going to get. Does that make sense?
Yes, sir.
Okay.
So can you please repeat?
See, it is the same neurotransmitter, right? It is, we are talking about acetylcholine, one neuron secreting acetylcholine, but binding to nicotinic acetylcholine receptor, causing the other neuron to fire, right? This other neuron on firing again releases acetylcholine, but it binds to muscarinic acetylcholine receptor. So the response is different when it binds to nicotinic acetylcholine receptor and when it binds to muscarinic acetylcholine receptor. The response is different, right? So the deciding factor is not acetylcholine here. The deciding factor is what kind of a receptor it is going to bind. Is the point clear now? Yes?
Yes, sir.
Okay. So in the similar fashion, now again one more neuron secreting acetylcholine binding to nicotinic acetylcholine receptor, making this neuron to fire. But this is going to synthesize dopamine and hence is going to secrete dopamine. Now dopamine receptors are again different and going to cause another different story of responses. Okay. Now a completely different set. Now till now we have talked about all sorts of neurons which are going to get fire on binding with acetylcholine. However, in adrenal gland, there is another kind of a response to acetylcholine. It binds to nicotinic acetylcholine receptor, but this neuron doesn't fire. It is kind of an inhibitory for secretion of epinephrine. Are you getting my point? So the difference here from all other ways or mechanisms is that here you're again releasing acetylcholine. It is again binding to nicotinic acetylcholine receptor, but the neuron is not firing. Are you getting the point? Okay. It it is responsible for secretion of epinephrine, this neuron. So otherwise, this neuron will always continue to secrete epinephrine. But as soon as the acetylcholine is released, it will stop secreting as epinephrine, that is adrenaline. So this is a kind of a negative way of regulation. So otherwise it's on, that means you are continuously going to get epinephrine. However, as soon as there is acetylcholine here, then your epinephrine secretion is blocked. Are you getting the point?
Yes, sir.
Okay. So now the response is dependent upon the postsynaptic neuron. Now the entire thing that I actually wanted to convey here is that what kind of a response you are going to get is actually dependent upon the postsynaptic neuron, how is going to react with the acetylcholine molecule or any other neurotransmitter molecule which is going to be secreted in the central nervous system. So now, in general, this is the way in which the nerve impulse actually travels. The first step is the neurotransmitter is released into the axonal terminal. Then the dendrite of the next neuron has receptors. Now we have talked about the receptors and the variety of way these receptors can respond to the neurotransmitter. And depending upon there is either stimulation or inhibition. We have already talked about this. And then you can actually cause the generation of an action potential in the dendrite, depending upon what type of a receptor the neurotransmitter is binding to. Now there is one more interesting facet to this, and that is about that how this entire mechanism actually happens. So let's take a small dip into it, and then perhaps the thing is complete, meaning in terms of your neurobiology section, what has been associated with your syllabus. So what happens? Let's take it into a pictorial view so that you can understand what I'm trying to convey. So now just imagine that this is the cell body of your neuron. These are the dendrites coming out from the cell body. Okay, and now it ends with an axon terminus here. So there are axon termini here with axonal bulbs that we have to see somewhat like this. Okay, so now you have the ends. Now we know that how now the neurotransmitter in the previous situation is going to generate an action potential and all that. When an action potential is generated, sodium ions get in. So now there is an inflow of sodium ions, and this continues all throughout the depolarization of the membrane till the axon terminus, right? Interesting thing is at the axon terminus, there is a arrangement which looks somewhat like this. Okay, where the synaptic vesicles are already docked. So this is a synaptic cleft. So this area is the synaptic cleft. This is the membrane of the axon terminus. This area of the axon terminus, and in this area you have these important proteins. These are the docking as well as calcium ion sensing protein. So these actually are involved in docking of the synaptic vesicles. Now, docking is basically they are immobilized. The synaptic vesicles stay here, but they are not going to release anything. Okay. So now what happens is as soon as this action potential, which is the depolarization of the plasma membrane, emerges here, these ion channels are basically calcium ion channels and they are voltage gated. So now they are going to respond to the voltage change, which is happening here because of inflow of sodium ions, right? Now, because of inflow of sodium ions here, there will be change in the membrane potential, and that will lead to voltage change. This causes opening of calcium ion channels. So calcium ions are here pretty much higher in concentration outside in the synaptic cleft. They are at higher concentration. So what happens is that calcium ions get in, start to rush in down their potential gradient. So they will always try to get inside the presynaptic membrane. Once they get inside, there is a building up of calcium ion concentration inside. Now, what happens with this calcium ion is these proteins are, that means the proteins on which the synaptic vesicle is docked, they are calcium ion sensitive. These are calcium ion sensing proteins. So as soon as they bind to calcium ion, so as soon as calcium binds here, these proteins tend to change their conformation. That means previously they were aligned like this. Now, as soon as calcium binds, they move apart from each other. They move apart. Now, what will happen if they move apart? Yes. Yes. What will happen if they move apart? Anyone? Okay. Till this point, are you with me?
So can you please repeat?
What
Sir.
portion you want me to repeat?
So the question.
Question is, see, okay, if you are paying attention, you should also pay attention to the question, right?
So I'm facing network problem. That's why.
Okay. Okay, but that actually leads to some kind of a repetition for other students as well, right? So you can actually refer back to the YouTube channel and you can see the lecture there properly in case you are facing network problem. Anyways, I will just repeat it for you. What we have talked about is that the membrane potential is changing because inflow of calcium, inflow of sodium ions. That leads to a voltage gated calcium ion channel opening, which causes
Okay,
an
sir.
inflow of calcium ions. And once the calcium ions get inside, then the calcium ion tend to bind to the two calcium ion sensing proteins onto which is docked your synaptic vesicle. As soon as now, previously, when there was no calcium ion, then these two proteins remain close to each other in close contact. And on their surface, they are carrying the synaptic vesicle. As soon as calcium ions start to build inside the presynaptic membrane, then these proteins start to sense, that means start to bind calcium, and they move apart. What will happen if they move apart? That was what was the question.
So they were blocking the neurotransmitter. Now the synaptic vesicles open and neurotransmitter rushes in.
No, exactly. It's exactly reverse of it. It's actually like this. Okay, I will show it to you. Now, these are the two docking proteins. Onto them is the synaptic vesicle carrying the neurotransmitter. The neurotransmitter is inside. Now, this is a presynaptic neuronal membrane. Okay. Now, what is happening here is that when calcium ion binds here, then they move apart. So, what exactly happens is this is the membrane of the synaptic vesicle, which is made up of phospholipid. And this is the membrane of the presynaptic neuron, which is also made up of phospholipid. It's something like, you must have seen this. I give this example very regularly. If there are small oil drops in water, then slowly what happens is that they coalesce together. That means they merge together in order to form a big drop of oil. Have you noticed this previously?
Yes.
Right. In the similar fashion, if this is lipid, the lipids tend to, what you can say, mix up together, or you can say merge together. And when they merge together, the internal content, which is aqueous, this is aqueous, that means it contains water essentially. The internal content is released in the synaptic cleft. Now this is the synaptic cleft, right? So you can see over here what is happening. As they move apart, previously what was happening, they were creating a barrier between the fusion of the two membranes, right? Now, as soon as the barrier moves apart, the two membranes can fuse. Make sense?
Yes, sir.
Okay. And so once that happens, you have released the neurotransmitter. Now the next step is that this calcium ion channel, which was open previously, is going to close now. Okay, because you have released neurotransmitter now. Now you have the synaptic vesicle, which is actually empty of neurotransmitter. There is no neurotransmitter now. But you don't want to lose this. See, the understanding is now the membrane, although it is phospholipid, but it is decorated with what you already know. These are myosin kind of motor proteins on its surface, right? So imagine now, if it completely merges with the membrane, that is the presynaptic membrane, then these will be of no use, correct? Are you
Yes, sir.
getting the point? Okay, so the myosin proteins will be useless now. If you want them to be useful again, you have to recycle the vesicles membrane back. So you need to convert it back into an empty vesicle which already has the myosin proteins which are necessary for mobilization of the filled synaptic vesicles. But the problem here is that we talked about this in the last class also. We talked about this in a different manner. Now you have myosin proteins. This is the synaptic vesicle. You have myosin proteins which are going to walk onto the actin cytoskeleton, which is forming the base of, or you can say as a track. It is going to use actin as a track, and myosin will walk on that track, right? So it is going to carry the vesicle, which is filled up here near the cell body, to the axonal terminus. Now here is the reverse situation. You will actually recycle the empty synaptic vesicles and you will transport them back to the area where you can refill those synaptic vesicles and retransport them back. This reverse transport of empty synaptic vesicles is called as retrograde transport. There is an anterograde transport where the synaptic vesicles are carried to the axon terminus. These are filled synaptic vesicles. And in retrograde transport, you transport the empty synaptic vesicles again back to the cell body so that they can get refilled. Okay, interesting?
Yes, sir.
Okay, and to do this retrograde transport, what do you exactly need? Sorry for this. Okay, to do this retrograde transport, what do you need? You need myosins, right? They are going to carry these vesicles back. So the interesting thing is this recycling is done by another protein, which is called as clathrin. And the clathrin coated vesicles, the myosins are blocked, right? And why do you need clathrin coating is because you want to block anything that is on the surface of the synaptic vesicle so that it should not refuse back, okay, by some or the other way. So you need to ensure this by, it is something like you have a polythene cover, okay, and that is done by clathrin. And once you have got your vesicle, empty vesicle back, then it is uncoated. That means the clathrin is degraded and you have an empty vesicle. Now you need to refill it. This refilling happens in an interesting manner. Okay, the refill needs, in order to refill, you need an antiporter protein. Okay, it's an antiport transporter, meaning it actually funnels out protons from the synaptic vesicle out, and in response to that it takes in the neurotransmitter. So this is your neurotransmitter which is getting inside. So protons go out while neurotransmitter comes in. Now if you want this continuous process to happen, you need to have a very high concentration of protons inside the vesicle, right?
Yes, sir.
Okay, and that is done by means of a proton pump which is ATP guided. So what is happening is that for every ATP hydrolysis, you are going to get protons pumped inside the synaptic vesicle, which is empty otherwise, right? So now this ensures one thing, what? That there will be always an inflow of the neurotransmitter. And there will be always an outflow of protons from the synaptic vesicles. So unless you don't have protons here, there will be no neurotransmitter getting inside. So first you need to activate the pump. The pump is going to pump up protons. So while you're doing retrograde transport, you are also going to utilize this for build up of protons inside the synaptic vesicle. And as soon as you reach to the end, that means towards the cell body, then there is a large concentration of neurotransmitters here, because the cell body is involved in its synthesis, and that neurotransmitter can get inside by using this antiporter, this proton neurotransmitter antiporter system, which will actually move the neurotransmitter inside the synaptic vesicle. So the synaptic vesicle becomes filled up with the neurotransmitter again and is ready to move back to the end and release the neurotransmitter again. So this is a cyclical process which continues over and over again. So do you have any questions regarding this process? Is the process understood clearly?
Yes, sir.
Yes, sir.
Good. So in case you are having any questions now, so now we have completed almost everything. We started from the architecture of brain. We started from understanding that how many different types of cells are there inside the brain, and then we figured out that how the nerve cell is involved in causing or in initiating the action potential. What is exactly action potential and how the depolarization of the membrane happens? It happens through what type of channels and what type of proteins are they, meaning how they control that, what kind of an ion they are going to transport, whether they are going to favor the sodium ion or potassium ion. And on that basis, it is decided whether the membrane undergoes depolarization. Once the membrane undergoes depolarization, that depolarized membrane is actually going to channelize that depolarization towards the end of the axon. And at the end of the axon, in response to that depolarization, there is opening up of calcium ion channels. And the calcium which gets inside is responsible for the fusion of the synaptic vesicle to the presynaptic membrane, which causes release of the neurotransmitter. This process is called as exocytosis. And then there is a recycling of the synaptic vesicle through clathrin coating, and this is called as an endocytosis mechanism. And then this empty synaptic vesicle is transported back to the cell body for its refilling with the neurotransmitter. This is the entire mechanism. And now, depending upon that, we also talked about that depending upon the neurotransmitter, what kind of response will be generated in the postsynaptic membrane. That essentially depends upon the neurotransmitter as well as upon the receptors which are present in the postsynaptic membrane. So this was all about how the nerve cell functions and what kind of biochemistry is involved in the functioning of the nerve cell as well as in transport of the nerve impulse. So do you have any questions? If you are having any questions, I am ready to answer. Or else this actually completes the section which is associated with neurobiology.